Jose Garcia Moreno. I am the director of the Academy of Catholic Thought and Imagination, and I welcome you to our technically second event of this series, the Monster Series. And I really want to thank you all for making this night possible, especially to Jeremy and Jane Brocker and Breno, who is here with us today, who will be one of the speakers in one of the future events from Monster, as well as Jane Brocker. So I'm gonna share very rapidly my screen just as a brief reminder for you and an invitation, a kind invitation to all of you to join us for the whole series. So let me just go very rapidly here. This is the Monster Series, which the subtitle or the login phrase for our event is "The Sleep of Reason Produces Monsters," which is something of a reference from Francisco de Goya in Capricho number 43. So there's some translations that have been said as "when reason sleeps" or "when reason dreams." And I really would like to very briefly read what was written, which is beautiful with the help of the group that put this event together. It says, "The last year has put to the test our ability to articulate the goals of science, freedom of speech, and society, and in so doing has brought to the surface several monsters that lurk therein. The themes that emerge include the growing demonization of the other versus the present need for social cooperation, the right to voice and the right to cancel that voice out, the objectivity of science in a world consisting of multiplicity of values and points of view that make up the Frankenstein-like social whole. With this in mind, we have decided to approach these four frameworks: science, which we're going to be presenting tonight, language, arts and culture, and society, with the hope of shining light on these issues and fostering an interdisciplinary dialogue. All of the films are online with your LMU credentials. You can actually watch them. The first one, obviously, is Young Frankenstein, which it is true, a very kind audience person actually remind me that Young Frankenstein is a black and white film, and this actually Mel Brooks fights very sternly against the studio that they wanted to colorize, and he fought for a black and white film. So we are sorry that we actually included a color representation of Young Frankenstein. Beautiful film. It's open to you. Just access the movie there. And today, obviously, Monday, October 25th, is Monsters in Science. Then tomorrow, we have Monsters in Language, and actually there are two sessions. One is at 8 o'clock in the morning, and the other one is at 2 in the afternoon. This one is a pre-recorded video presentation, and there's also this live online discussions via Zoom at these two different sessions. So please join. It's actually a very, very interesting presentation. Please join us with this live discussion. Let me go back, and then we have Monsters in the Arts, which is going to be hosted by Breno and Jane Brocker, and this is a live online presentation via Zoom. As you did with this event, please do your registration online. This is October 27th at 7 o'clock, so that is Wednesday. And then we have an invitation for you for a live event, which is Monster in Society, where Carla Marcantonio, Associate Dean of the School of Film and TV, and myself, we will be discussing the place of monster in filmmaking. And we are going to be screening live Akira. For those of you who have not seen it, is a must-see movie for all of you. And also we will have a short film, which is, we have the image here of Jonathan Glazer, which is a fantastic and frightening short film, which is The Fall. We will be showing that as a short film within the program, so a feature and a short. And there is, we are hosting a light reception before the film for just getting together and talking about monsters. So thank you so much for everything. Let me stop sharing that. Oh, stop share. And I'm gonna put you in the hands of Jeremy. Thank you so much, Jeremy, for allowing me to do this brief presentation.
Hello, everybody. I'm Jeremy Wasser, and I'm an associate professor of physiology in the Department of Veterinary Physiology and Pharmacology at Texas A&M University, in spite of the fact that I live in California, and I've been on the faculty there for a long time, and ran a research lab for many years, not actually in neurophysiology, in cardiovascular physiology, but I also teach neurophysiology to students, and I also teach the history of medicine, which is one of my interests. And one of the great things about being a physiologist is that we are trained as generalists compared to many other scientific disciplines. And what I mean by that is that because of the nature of the body, which is what I'm going to be talking about tonight, understanding one organ or one organ system, the heart, the brain, and not understanding and having appreciation of the anatomy, the physiology, and the complexity of every other part of the body is a meaningless exercise. You end up not knowing anything. What I tell my students when I teach physiology is that in a very, very real sense, in an anatomical and a functional sense, in the body of an animal, like a human or other animals, everything is connected to everything else. So the fact that I'm not technically a neurophysiologist did not deter me from taking on this assignment to talk to you about the neurophysiology, the neurobiology of fear. And in the week leading up to Halloween, that seemed like a good thing to do. So I'm going to go ahead and start the presentation. I'm going to start my screen share. I have a PowerPoint for you. And there'll be time at the end for questions and answers. And for some of you who have a physiological or biological background, some of what I'll say will be review, but for many of you, I think that won't be the case. And then I've burrowed through the recent literature looking for interesting and cool studies to talk to you about. So I think there'll be something here for everyone, regardless of what you already know about neurophysiology. So I will share my picture and sound with you. And I will go to Monster in Science, the Neurophysiology of Fear. For those of you old enough to remember the TV show, The Twilight Zone, that was, of course, the theme from The Twilight Zone, a very creepy, scary show. And then for those of you who are younger, there's been a remake. So you perhaps recognize that music. Now, one of the things that those of us who worked on this Monster Week did, and this was the work of our film colleagues, Jose and Carla, was put together a series of films that are the themes for the individual lectures. And the one for my presentation on the Monster in Science, the Neurophysiology of Fear, is David Lynch's The Elephant Man, which is one of the films that, if you have LMU credentials, you can see. And the predominant emotion displayed by the people in the movie and in real life on seeing Joseph Merrick. In the movie, it's John, but his real name was Joseph Merrick, the so-called Elephant Man, was fear, anger, and disgust. But these emotions didn't necessarily last. Not everyone felt that way. Dr. Treves, the doctor who in real life looked after the Elephant Man, looked after Joseph Merrick, did not feel that way. And feelings for him would change over time. So the questions that I wanted to provide you with information to think about and understand and assess are, why did people fear him? And what does it mean to feel fear? We've all felt fear. Everyone in this Zoom room has been frightened. What does it mean to feel fear? Where does fear and the associated emotions Where do they come from? What are the anatomical and physiological components of the fear response? And those are the kinds of things we're going to be talking about today. And I'll try to present you with a mini version of the state of the art on what's going on when we are afraid. I will talk about these biological questions now at the start of this presentation, but I'll come back to Joseph Merrick. I'll come back to the Elephant Man towards the end of the presentation, because I want to talk to you about his actual condition, which was not known at the time of his life. We now know what he had, and we know something about it. And I will talk to you a little bit about his specific medical case. But first, brains. So there's our Elephant Man. But first, brains. This is a still from Young Frankenstein with Igor holding a brain. We human beings have been interested in brains for a long, long time. In fact, the very first known surgical operation, the oldest surgical operation in the world, is trepanation, is the drilling of a hole in someone's skull. This was done, we have prehistoric skulls. We have 12,000-year-old skulls with these circular holes, which must have been drilled with a bone or a stone tool in some cave somewhere. And what the shaman or the doctor of that time, the healer of that time, was trying to do, although they didn't really know it, was to relieve intracranial pressure from a close skull injury. So someone was hunting mammoths and injured their head and was bleeding inside, and probably they thought this was some sort of demonic thing or religious thing, but these shamans would drill this hole. And if you're careful not to penetrate a layer of protective tissue that covers the brain called the dura mater, if you're careful not to penetrate that, you can survive that operation even 12,000 years ago. And we know this because we have skulls in which those holes have healed. So it's quite extraordinary. In ancient Egypt, where they had a rather sophisticated medical tradition, on the other hand, they thought brains were not very important. They were the only organ not preserved. In the embalming process, they were taken out through the nose with a big hook and they were thrown away, while everything else was carefully preserved. Forgive me for the medical historical sidebars. I can't help myself. If we move to the 16th century, Leonardo da Vinci, among his many interests, was human anatomy. He was a superb human anatomist. And the picture on the right is from his many pages from his sketchbooks, and it shows the cerebral ventricles, the spaces in the brain in that top figure that are filled with cerebrospinal fluid. Leonardo did not know what their function was, but he knew that they were there. And then in both of the human head figures, you can see the cranial nerves that leave the brain and go out to the body and are part of the autonomic nervous system, which I'm going to talk about in just a minute. The other thing that's curious about this picture is that the bottom exploded view of the human head, skull, and brain. This is the first representation we have in anatomy drawings of an exploded view, which is now, of course, quite common. And Da Vinci was the first one to do this. The picture on the left is from Andreas Vesalius's famous work from 1543, and that was the Humani Corpore Fabrica Libri Septum, the 7 books of the human anatomy, the first really modern anatomy textbook. And you can see the beautiful images there of the human brain. And we will talk about this and show you some brains as we move forward in this presentation. But we need some anatomical and physiological background. And that's what I want to present to you now. This is a picture I show my students when I teach neurophysiology. And what it shows is the breakdown, the anatomical breakdown of the parts of your nervous system. It's not just your brain. In fact, the brain and the spinal cord are part of what we call the central nervous system, or the CNS. So any nerve cells, any neurons, nerve cell tissue that's contained within the skull or the spinal cord is part of the central nervous system. But outside of the skull and the spinal cord, we have lots of nerve cells. A nerve, by the way, is a bundle of many nerve cells. When you see a nerve, when you make a dissection, you're not looking at a single nerve cell, a single neuron. In fact, you're looking at probably hundreds or thousands of them in a cable that goes out to the body or back to the central nervous system. So if you look at the left side of this diagram, we have what's called the afferent division. These are nerves that come from the body and go back to the brain and spinal cord. So you can see below in the little yellow boxes, they are picking up sensory stimuli from things like the eyes, the ears, the nose, pressure receptors in the skin, pain receptors in the body, and so on, temperature receptors, and visceral stimuli, which are signals coming from your viscera, from your guts, from the inside of you, that are going back to the central nervous system, carrying information from the body to the brain. Then on the other side of this loop, we have the efferent division, which are nerves leaving from the brain and the spinal cord and going out to the body. And there's a division of labor in this anatomy as well. The so-called excuse me, somatic nervous system goes to our skeletal muscles and allows us to move our skeletal muscles. The autonomic nervous system is a almost always unconscious system and is made up of two parts. And I'm a little sorry about having to go into this much detail, but it will come back to haunt us later in the talk. The two divisions of the autonomic nervous system are the sympathetic and parasympathetic nerves. And these go, as you can see in that bottom square, to other kinds of muscle, smooth muscle, the cardiac muscle, your heart, to glands that secrete hormones and other substances, exocrine and endocrine glands, and are very, very important. And then finally, and I won't talk very much about this today, although it actually is involved in the physiology of fear, as is everything, it turns out, in our bodies practically, there is a nervous system in your gut. In your gastrointestinal tract, we have what is commonly referred to as the little brain. So if you ever thought that you were being governed by your stomach, by your urge for a donut or something like that, you were right. You were. The enteric nervous system, or the little brain in the gut, is connected to the central nervous system via the autonomic nerve. So as I said in my little intro about physiology, everything in the nervous system really is connected to everything else, but the function of these different pieces have been distributed. You have different parts that are doing different things. And we'll look at some of these in more detail right now. So here we have another picture from a physiology textbook showing the parts of the brain you see when you just open up someone's head, a human being's head, and look down. And those two big gnarly brown parts are the two cerebral hemispheres. They are the cerebral cortex, which in human beings is very large and very, very wavy. It has these folds called gyri and these spaces called sulci, and they're there to create even more volume and surface area. We have big brains, but there are important subcortical parts of the brain, some of which are shown in red in this diagram. The limbic association cortex, which is involved with emotion, which is important, of course, in the physiology of fear. The thalamus, which is there in about the center of the brain, which is an important sort of way station for signals. I'll show you some pictures coming up. Information comes in from the environment, from outside the body, through the sensory system, gets to the thalamus, and then is sent by the thalamus to other parts of the brain for integration and interpretation. The hypothalamus, an important part of the brain for memory and learning, and the amygdala, which I will talk about a lot tonight. The amygdala there, that little round thing, basically almond shaped, and it's bilateral. There were two of them, one in each hemisphere. And the amygdala is absolutely critical for the emotion and sensation of fear. It isn't the fear center exactly, that's not a way to think about it, but the activation and involvement of nerve cells in the amygdala, as you will see, is critical for one to feel fear and to process and integrate fear. And then finally, just because it's cool, you see the little antennae in the front there, the olfactory bulbs. It looks like this person has a little antenna. Those are part of what we call the olfactory cortex, the part of the cerebral cortex that interprets signals from your nose, olfactory signals from your nose. And that's actually also involved in the sensation and processing of fear. But in general, humans are not, we don't smell well. I don't mean we stink. I mean, we don't have a good sense of smell compared to animals that rely more on smell. Human beings are primarily visual animals and devote a large part of the cortex to vision. But animals like dogs, animals like turtles, actually, which I studied for many years, have an enormous olfactory bulb because they need more brain to process all that information. So as I said, you are getting signals from all over the body that are going back to the central nervous system as part of the fear response. And this has been taken advantage of by the makers of horror movies. Most of the signal from a horror movie, or a scary movie, I should say, is of course visual. and acoustic. You see it and you hear it. But so a very clever guy named William Castle, in a grade Z horror movie from 1956, I believe, involved another sensory dimension. And this is a movie called The Tingler. I'm guessing you haven't seen it, although it is a classic cult movie. And in The Tingler, it's about this parasitic monster that lives inside of people, and you know it's about to get you or climb into you and parasitize you when you feel a tingling at the base of your spine. And what William Castle did in selected theaters, because back then there was no Netflix and no streaming and you had to physically go to the theater, he had a device built into some of the seats that would vibrate. And the only way to fend off the tingler was to scream. And so people in the movie would be screaming and people in the chairs that were vibrating would be screaming. And if you value your life, you will also scream. This was very clever, and it was taking advantage of a component of the neurophysiology of fear, which is that it's totally multifactorial. It involves essentially all of the senses. It involves pain reception. If there's pain involved, it involves pressure, mechanoreception, which is the example of the tingler. And of course, it involves vision, it involves hearing, it involves olfaction. It's what makes it complicated. And there've been a lot of studies, and we still don't entirely understand what's going on. So I already said that nerves are made up of individual nerve cells called neurons. And I just wanted to show you this picture because we'll be talking about this relationship a little bit later too, when we start looking at what's going on inside the amygdala. So here we have two of these nerve cells, and they're connected to one another. So what's happening in the nervous system is a signal goes from this upstream neuron down through this piece, which is called the axon. And some axons have these little sausage casings on them called myelin, and some don't. And then when it gets to the next neuron downstream, it doesn't physically touch it. This was not known until the late 19th century. It was uncertain by physiologists of that time how nerves connected to one another, how nerve cells connected to one another. But it turns out there's a space there. And this space is called the synaptic cleft, or this piece of anatomy is called the synapse. So what is happening is the signaling in a nerve cell is electrical. So the electrical signal starts up here in the cell body. It goes down the axon, this piece is called, and when it gets out to these axon terminals, the signal causes a release of a chemical. And that chemical, which is called a neurotransmitter, diffuses across that space and binds to special chemicals on the next neuron in the chain and activates it or inhibits it. It depends on what chemical is getting released. So signaling in the nervous system is electrical and it is chemical. Now, electrical signaling in biological systems is a little bit different. It's not like in a wire, but it is a current. So we have an electrical signal, and then in between the neurons, we have a chemical signal. And what these neurotransmitters do, they play a very, very important role in regulating what the next neuron is going to do. And of course, in your actual nervous system, it's very rare, almost unheard of, for one nerve cell to be connected to only one other nerve cell. In fact, many nerve cells, particularly in the central nervous system of the brain and the spinal cord, are connected to hundreds or thousands of other neurons by these synaptic connections, which means signaling is going to spread out. It will converge, it will diverge, and spread through the brain from one part of the brain to another. And these parts subsume different important functions in the interpretation of signal coming from the environment, in the processing of that signal, and in deciding what you as an individual are going to do about the signal. Are you going to fight? Are you going to flee? Are you going to laugh? Are you going to cry? Are you going to scream because of the tingler? What are you going to do? And then finally, one last picture from sort of conventional physiology books. I mentioned the autonomic nervous system. It's very important in our discussion tonight. And this is a schematic diagram of it. It's showing the nerves that leave the base of the brain. Those are the cranial nerves. showing the nerves that leave the base of the brain and go out to various sensory systems like, or come in from sensory systems like the eyes, the ears, and the nasal mucosa. And then running down your spine, you also have nerves that leave the spinal cord and go to everywhere. That's kind of the thrust of this picture. They go to your lungs, they go to your heart, they go to your GI system. The two divisions, the sympathetic division of the autonomic nervous system is generally excitatory. It excites neurons and makes them fire. The parasympathetic division does the opposite. It inhibits signal and makes things be less active, with the exception of the gut. In the gut, the sympathetic nervous system inhibits and the parasympathetic stimulates. This is important because, as we'll see in a moment, when you are frightened, when you initiate what we call a fight or flight response, you activate the sympathetic nervous system. And what we mean by that is you send lots of signal down those neurons, and those nerves then leave the spinal cord, go out to the tissues, to the heart, to elsewhere, and ramp that stuff up so that you can do what you need to do to avoid whatever it is you're trying to avoid. When you're doing this, the last thing you want to be doing is digesting food. So in fact, your body will inhibit what's going on in your gastrointestinal system and redirect its resources and energy to the parts of the body you actually need if you are trying to deal with a frightening situation. So now, after this sort of basic overview of how your nervous system is put together and a little bit about what's going on in it and how neurons work, we need to move on and talk about fear.
I'm not afraid.
Yeah. You will be. You will be.
Yes, you will be. So, fight or flight. In fight or flight, we have a balance between those two branches of the autonomic nervous system. The parasympathetic, the guy lying down, we sometimes say it dominates during resting and digesting, rest and digest. And then fight or flight is when we activate our sympathetic nervous system. We got the guy running away, and he's running away from a snake. I like this picture because there's actually a literature on whether human beings have an intrinsic, hardwired fear of snakes. It isn't known, we're not sure. Lots of people are afraid of snakes. Most of that fear is learned, but it may be that we are intrinsically afraid of snakes. But in any event, when most of us handle snakes, I actually did my PhD work on snakes, so I'm not actually that afraid of snakes. They were not poisonous snakes, however. But most people, a snake will activate the sympathetic nervous system, and that is our fight or flight, where the sympathetic parts of the autonomic nervous system are dominating. Now, a much more complicated diagram that I'm not going to walk you through, of course, from the literature on fear is this paper. This paper was called The Biology of Fear and Anxiety-Related Behaviors. And what I want to point out are the parts of the brain and the interconnectivity of this system. So here in the middle, we have the amygdala. The amygdala has more than one part. It has a basolateral amygdala, a central nucleus of the amygdala. The BNST is an important part of this system as well. And then we have our thalamus, which I talked about. That is a routing center. You can see all the arrows going to it and going from it. And sensory information comes in from the outside. Sensory information comes in, and it goes either directly to the amygdala or through the thalamus. The hippocampus is involved. There are these neurotransmitters illustrated in this figure: glutamine, noradrenaline, and another one called CRF. GABA, gamma-aminobutyric acid, is involved, and other parts of the brain as well, the hypothalamus. And you can see during sympathetic activation, you will have a tachycardia. That's an increased heart rate. You'll have an increase in blood pressure. All of these things are designed to get you ready to do one of two things: to flee or to fight. And if the parasympathetic system is activated, which it also sometimes is in fear, you will have freezing or escape behavior. So many animals, including humans, will sometimes freeze when they are exposed to certain kinds of fear stimuli, particularly fear stimuli where the location of the danger is uncertain. One of the natural responses of many animals, of rabbits, for example, of rodents, but also to some extent of human beings, is to freeze and then try to assess where that danger may be. That freezing is being mediated through the parasympathetic division of the autonomic nervous system. Panting and respiratory distress, we see this in some animals. The startle response, which I'm going to talk about, when you have a sudden, what they call in horror movies a jump scare, where the zombie comes out and you're not expecting it. There's a startle response. And also in some cases, urination, defecation, and bradycardia. But part of what I'm trying to get across with this figure is the absolute complexity of it. And of course, what also happens is as the signals leave your nervous system during a fear activity or a fear response, you're activating other parts of your body. As I said, the heart, of course, the respiratory system, the lungs, and also the endocrine system. So hormones are leaving, particularly hormones from the adrenal gland, such as epinephrine. And these are all part of what's going to give you that final response to this particular stress. Just another picture, a real picture of the brain, showing you where the amygdala is located. This is a transverse section stain. The amygdala is located in this region, and here's a photomicrograph showing you the brain, the amygdala, and some of the divisions of the amygdala, and then a cartoon, the basolateral part, the lateral amygdala, some ancillary regions of the amygdala, and then there's even a piece called the extended amygdala in the BNST. But the question really is, once we realized that the amygdala was important in modulating emotions in general and fear in particular, it's something we wanted to study. And back in the day, the way this was studied by neurophysiologists was by ablation studies. And what that means is they would take an animal, a research animal, not a human, but a rabbit, a cat, a rodent, and they would destroy regions of the brain and then see how the animal behaved. And it sounds terrible, and kind of is, but this was how that research was done, and we learned a great deal about what the different parts of the brain do under different conditions from these so-called ablation studies. The other way it was studied was more humane. There are human beings who, through disease or injury or through trauma, have parts of the brain destroyed. And these individuals will also be studied to see what their responses were, physiological and behavioral, in the absence or with compromised regions of the brain. And from this, we learned a great deal about what each part of the brain is doing. We now have a way to study this in vivo, in a living human being, a conscious human being, without probing their brain with anything nasty. And I want to talk a tiny bit about that because I'll show you some fear studies using this technique. The technique is called functional MRI. Now, some of you may have had an MRI scan, a magnetic resonance imaging scan. And if you have, it probably was not an fMRI, it was probably a regular MRI. And this is a guy, and you can see he's smiling. He's a happy camper because he knows that when he gets put inside this giant, powerful magnet, his body is not going to be zapped with X-rays. It is different than a CT. It's different than an X-ray machine. It uses a powerful magnetic field and a short radio frequency pulse to derive imaging data from the body. And I've actually done a magnetic resonance work myself, not so much with imaging, but with a sister technique called magnetic resonance spectroscopy. And it is really kind of magical because you put your specimen in the magnet, you do the technical things you need to do, and you get a picture or you get a readout of the chemicals inside the structure while it's living and beating and thinking and working. So if we put this guy in this magnet, we can get a series of images that look like this. And we're basically taking slices through his brain without having to slice through his brain. So it's not a horror movie. It's a painless and extremely effective and quite amazing medical imaging technique, and used in conjunction with other techniques. It's routine in hospitals now. And you can actually look, as I said, at different depths of the brain. You can look at slices through the brain and look for anomalies, look for things like tumors, look for abscesses, look for regions of the brain that are not doing well. But in fMRI, in functional MRI, you're able to make an image of the person's brain and then have them engage in an activity. For example, watch a scary movie. And this has been done in an MRI magnet. Or be exposed to a scary stimulus. There was a study done in which a tarantula was moved closer and closer to the subject's feet. And they knew this. They could tell that this was happening while they were in the MRI. You take a before and an after picture, and you can use this technique to see which regions of the brain are activated, which regions of the brain light up, so to speak. And the reason they're lighting up is because when you use a part of your brain, the metabolic rate of that part of the brain goes up. And when the metabolic rate of the brain goes up, its oxygen consumption goes up, needs more O2. And when the brain needs more O2, it needs more blood. because that's where the O2 is, inside the blood. So when you do a functional MRI study, what you see are the regions of the brain that are getting more blood flow at that moment, and it is because they are activated. So one can do studies using fear stimuli and look and see what parts of the brain are in fact involved and what they are connected to. And I want to talk to you a tiny bit about this amazing recent study from 2020 by a group of Finnish neurophysiologists published in the journal Neuroimage. And it has a funky science title, The Sociable Neural Systems for Unconditioned Acute and Sustained Fear. But I call it the Finnish horror movie study because that is what it was. They surveyed their subjects and asked them, what are your favorite top 10 scariest movies? And they came up with this list of 10. Now, one of my colleagues, Tani Carrillo, helped me put this presentation together, is actually an expert, an authority on horror and horror movies. And she said, this is the lamest top 10 horror movie list of all time. But nevertheless, this is what those Finnish subjects said were terribly scary movies. And in particular, Insidious and The Conjuring 2. And we know this, and the researchers concluded these were the ones to study, because if you look over on the right-hand side of the table, the jump scares are 24 in Insidious and 22 in The Conjuring 2, and much, much lower in the other members of the top 10. So they showed clips of these movies in a specially modified system to subjects in the MRI. And what they got were pictures that looked like this. Now, the top images are showing a jump scare, and there are a lot of regions of the brain lighting up. The amygdala is lighting up. That's AMY. The parahippocampus is lighting up, which is near it. So here are the amygdala on both sides lighting up, and there's a lot of cortical regions lighting up as well in the jump scare, and in the what they call the sustained fear experiment, in which the part of the movie was one in which you knew something was going to happen. The music cued you in, or for example, one of the characters in a movie is climbing the stairs to the attic after his or her colleagues told him or her not to go. Well, when that happens, you know that person's dead, pretty much. You know the zombie or Freddy or Jason or somebody is going to get them. And this would generate a different kind of fear response in you, the viewer. It would be so-called sustained fear response. And what this study demonstrated was totally different regions of the brain light up. There's a lot less activity in the amygdala, and there's a lot less overall cortical activity. So it's really quite amazing about that, and really quite an extraordinary thing. In a different study, not the Finnish horror study, but in a different study, these researchers were interested in seeing what scary faces, the response of subjects to what frightening faces. It was known already that when you see someone who is frightened, and you recognize that expression even if you don't know what's frightening them. And fear is in fact contagious. When you look at someone whose eyes are wide, whose mouth is slightly open, who's showing a little bit of teeth, this is the classic fear face response. And you will begin to be fearful as well. And what these researchers did was they looked to see whether there was any correlation in the perception of fear faces based on where you are in your heartbeat cycle. So this is one of these connectivity things I was talking about. Your heart beats and relaxes. I'm a cardiovascular physiologist. I could talk more about it, but I'm not going to. Basically, your heart contracts and pumps blood and relaxes and fills with blood. And contraction is called systole and relaxation is called diastole. And you can look at that and monitor it with an ECG, like over on the left. And what they looked at were these emotions: fear, disgust, a neutral feeling, and happy. They showed people faces that showed fear, that showed disgust, that were neutral, didn't show anything, and that were happy faces. And they looked to see how people responded at different points in the cardiac cycle. And it turned out you are much more sensitive to fear, in particular, when the heart is actually beating. And they also looked at the response of the brains of people to fear. And they had controls. And then they had another group of people who suffer from a condition called it's an autonomic nervous system problem. It's called pure autonomic failure, so PAF. In pure autonomic failure, there is a failure of the autonomic nervous system, not a complete failure, but there's a failure in those nerves of the ANS. These people have problems regulating blood pressure, they have problems regulating temperature, It's really quite a serious problem. It's an example of what are called dysautonomias, which is a fancy word for a failure of the autonomic nervous system. And the images from fMRI are from the patients with PAF. And what they're illustrating in that circle are amygdala and insula, which is another brain region associated with fear. They're lighting up, but they do not light up nearly as much as in the case of controls. And then the graphs on the right show this, that the response of controls is generally speaking much higher than that of the people with pure autonomic failure. So fear is not just in the brain, it's in the peripheral nervous system as well. And as I've already said, it involves every organ of the body in very, very complex and fascinating ways. We also know from studies that there's a couple of ways that a fear stimulus gets into your brain and starts to get processed. So here we have a fear stimulus. I actually chose that picture. That's a green tree viper, very poisonous, very dangerous, and very scary. And that fearful input can either go from the thalamus, that's the purple thing in the middle there, directly to the amygdala, or it can go from the thalamus to the cortex and then to the amygdala. That's the piece in the back. That's the visual cortex in this example. The part of the cortex that actually allows you to see. Without that part of the brain, you're blind, even if your eyes are in perfect condition. What this is telling us is that there are two pathways for a fear stimulus in the brain. One is direct to the amygdala through the thalamus, and one is routed through the cortex. We call the direct one the low road, and we call the one through the cortex the high road. And you respond differently depending on whether the signal is going through the high road or the low road. So this is a representation of your perception of a scary face, of a scared person's face on the low road. And this is all activity in the brain below the cortex, below the cortical part of the cerebral hemispheres. And then if that signal is routed through the cortex, you get what looks like a drawing, but actually it's a much more detailed view. You can see that the eyes are wide open. You can see that the mouth is open and the teeth are showing. There's far more information on that right-hand picture. And it is in fact going to be in some ways a scarier picture. The reason why we have the two roads apparently is that the low road processing is much quicker. So what these researchers in these kinds of studies have demonstrated is that you can be fearful without actually feeling fearful. You can begin to have a physiological fear response without a conscious sensation that I am scared, which I think is fascinating. In most instances, we do process these things cortically, and then we know that we are scared. But the physiological responses to fear begin before that happens. And we think the evolutionary value of that is it's quick, it's fast, and it begins to get the body ready to do something about the fearful stimulus. Now, I want to show you a GIF. Animated GIF from the Elephant Man. This is the so-called reveal, where the poor nurse walks in unsuspectingly on Joseph Merrick and sees him. And look what's happening. Her eyes are going wide. Her mouth is going open. Her right hand is trembling. That is actually something we would expect with a lot of sympathetic nervous system stimulation. And she's dropping the bowl of oatmeal. It's a classic fear response, and it is one that has been processed through the cortex. The other part of this that's critical is the importance of the eyes. And there's been quite a bit of research done that have looked at the eyes and the importance of them. One of the things that's scary about Darth Vader, is the suit and the voice and the fact that he's a Sith Lord and he's bad news. But they could have made that headpiece really differently. They didn't have to have the bug eyes. And it always struck me, as someone interested in animal biology and insects, that he looked a little bit like a praying mantis, which is also kind of a scary and a very predatory insect, and to other insects, a very dangerous one, not so much to people. So what's going on with the eyes is very, very important in terms of our perception of a fearful stimulus. And then I just wanted to show you this because it's funny. This is from another 1950s grade Z horror movie. This is from the movie called The Brain from Planet Eros from 1957. And it's an alien that looks like a brain with eyes that tries to take over the world, but is defeated by a policeman brain from the same planet who comes to save us. So eyes and brains, very, very important. Now, I want to talk a little bit more specifically about the amygdala and the high road and low road. This is also a somewhat complicated figure. The paper said it was a simplified figure, but it's fairly complicated. But the points I want to make are here in the middle. We have our amygdala, we have the lateral amygdala and the central nucleus of the amygdala. And here we have low road signals coming in, either from, in this example, from hearing, acoustic signal, or from actually a pain signal. And that processing begins already, and you will already begin to physiologically respond. So there'll be signal coming out of the central nucleus of the amygdala and going to cranial nerves, to the BNST, to other parts of the brain, even if you don't know that you're scared. But signal can also go from the acoustic and pain receptors to the brain via the thalamus, and from there to the lateral amygdala, and that is the high road, where you are processing already and bringing into consciousness what is going on. And of course, in most fear activities or fear episodes, both of these things are happening. And you can see in the text at the top of this slide, the kinds of things that are going on. Open mouth, lower jaw, facial expression of fear that's mediated from the cranial nerves 5 and 7, activation of the sympathetic nervous system from the hypothalamus, increased respiration and so on, activation of secretion of stress hormones, ACTH, adrenaline, and the other important stress hormone, cortisol, freezing. That's a somatomotor effect. That's where you actually stop using your muscles to freeze. Adrenaline goes up, cortisol goes up as well. Exaggerated reflexes to sudden and threatening stimuli. So what we saw the nurse doing was an exaggerated reflex. She jerks her hand and drops the ball. Another thing you may have experienced yourself when you're scared is you move your arms to your body in particular ways. Now, in a movie theater, when I'm scared, I do this. I want to cut off the visual stimulus and reduce the amount of signaling to my amygdala, because Germans would say a Weissei. I'm a soft-boiled egg. I actually don't like scary movies that much. But the other thing you see people do when they are scared is their arms come up. And this is actually defensive posture. This is actually something that is actually priming you, even though you're not thinking about it, for either fight or flight. And then there is also facilitation of the cerebral cortex. That increases the degree of arousal. There's another part of the brain called the reticular activating system, which is fundamental in your consciousness state. How awake are you? How aware are you? We want to be very aware in a potentially dangerous, frightening situation. Among the things we see, by the way, is something called mydriasis, which is an enlargement of the pupil. So your pupillary response, your pupils open when you are frightened, even if you're not in low light, because your body is trying to bring in as much light as possible so that you can see optimally and hopefully identify what it is that is threatening you and do something about it. Now, from another really nice paper that reviewed this information called Understanding Emotions, Origins, and Roles of the Amygdala, I just want to show you a couple of images of a cartoon of the amygdala showing the different parts. But what I want to emphasize here is the flow of neural traffic, the flow of neural signal, and what's going on with information. And here we have over on the left, signals coming in from the senses, from the body, processed through the thalamus and going to the lateral amygdala. And there they are synapsing, they're connecting with neurons in the amygdala, and ultimately with neurons at the top, those are the green ones, that leave the amygdala. Some of these neurons release a neurotransmitter called glutamine. They're called glutaminergic or glutamergic systems, and that is an excitatory neurotransmitter. It makes the next neuron fire. Others are GABAergic or GABAergic neurons. They are inhibitory. They make the next neuron in line fire less. And it is the balance of these that determines how you feel and how you will respond to fear. So if we look at that, in this figure, we see this is in a person who is not afraid. This is in a standard situation. You're listening to my lecture. You're not intimidated. There's not going to be a test. You're not worried. And what you see is that signals coming in through the excitatory neurons, the glutamate ones. There is some inhibition from the GABA ones, and the little starbursts indicate a normal firing rate, a normal degree of stimulation. And they're flowing through the different parts of the amygdala and then are leaving it. And you can see the synapses and the neurotransmitters represented here. If we look instead at someone who is fearful, who's been subjected to a fearful stimulus, we see something different. We see a decreased firing of the inhibitory fibers. That's up, the green ones on the upper right, they have smaller little starbursts, and an increased firing rate of the glutaminergic fibers, the excitatory ones. Let me bounce back and forth if I can, because sometimes that makes it easy. Normal, inhibited, normal, that's inhibited, that's normal, resting, scared, not scared, scared. So when you are exposed to a scary thing, there are changes in the amount of signaling coming into the amygdala, and the balance that you achieve ultimately is this balance of the stimulatory neuronal effect and the inhibitory neuronal effect. And then that signal leaves the amygdala, goes to the cortex and elsewhere, and determines what you're going to do and how you feel. If we look at it in terms of information flow, this is what's happening. There's an inflow of sensory information there on the left. There's implicit emotional learning and memory. Fear can be learned and remembered. It can also be, in a sense, forgotten. We call that fear extinction. Then there's processing and recognition, integration of sensory information. That takes place in the basolateral amygdala. From there, we have learning, fear conditioning, and we have information filtering. How dangerous is this situation? Security versus danger. Because again, in the natural state, we don't want to be fleeing from things that are not scary, because that takes energy. And you want to conserve your energy for important things like finding food and reproduction and stuff like that. So you don't want to be scared all the time. You don't want to be fleeing or fighting inappropriately. And that involves assessing security versus danger. There's a transfer of the final situation assessment to the central nucleus. And from there, at the top of the figure, it goes out. Autonomic and endocrine physiological responses. And then from the BLA, we have behavioral responses and decision-making via cortical inputs. Now, I told you earlier that one of the ways people have studied this is by looking at human beings who have damage or are lacking key structures in their nervous system. And there is a person, we only know her by her initials, they are SM. And the SM is a woman. She was 44 years old when she was first studied, and she has bilateral lesions of the amygdala. She is basically missing both parts of the amygdala. She doesn't really have a functional amygdala at all. And this is another one of the movie fear studies in this clip. It's fear induced by film clips. And they compared people with normal amygdalas, and they compared the patient SM. And you can see the maximum amount of fear as assessed by how they responded by skin conductance, which measures sweating, because you sweat when you're scared, by cardiac measurements, and also by personal reporting. You can see she wasn't scared. She's been called the woman without fear. And in her own personal history, she's had some very scary things happen. She was assaulted at night at knife point. And when reporting this in these studies, she said she understood it was dangerous, but she was not fearful. And this is because if you look at an image of her brain, there are two black holes where her amygdala should be. There are other patients with a disease called Ullrich-Weier's disease that have calcium deposits in their amygdala. And depending on the degree of calcification, they exhibit differing degrees of an attenuated fear response. Well, what I want to do now in getting towards the end of this talk is bring you back to the reason why we're here this week before Halloween, to the theme of our monster workshop. And as Jose said in his intro, our phrase from Goya is the sleep of reason creates monsters, which my interpretation as a physiologist is when reason sleeps, when the cortex is quiescent, we create monsters and can still be fearful. And you now know from what I've talked about tonight, that we can be fearful without knowing that we are afraid. It doesn't require higher cortical processing, but it often does and can. And it involves many, many parts of the brain talking to one another back and forth, this interconnectivity, and it involves the rest of the body. And that includes things, again, like sweating. It includes things like what's happening with your eyes and your mouth. It includes goosebumps. It includes the phenomena of Gänsefleisch, the Germans say, goose skin, goosebumps, where the little, what they're called the piloerector muscles at the bottom of each hair follicle are stimulated. They are a special kind of muscle called smooth muscle, and they are stimulated by the autonomic nervous system. So when you are frightened and you have a high degree of sympathetic stimulus, those muscles are triggered and your hair stand on end and your skin makes that little dimple. Now in humans, this isn't doing a whole lot because we don't have a lot of hair. But if you're a fuzzy animal, like a dog or a fox or a wolf, and you do that, you double your size. You start to look huge, which is the intent. And this is a response to a fearful and potentially dangerous situation. Domestic dogs will do this if they're not well socialized. If they're well socialized, they don't do it. And that is also part of this integrative fear response. And integration is what physiology is all about. It's about integration and it's about regulation, homeostasis. And that's what's going on with fear. So finally, another clip from the movie with Anthony Hopkins as the surgeon Treves. This is when he first displays the Elephant Man to his colleagues before he begins to, I think, feel empathy to get over his initial emotions, which were complex, but which involved some fear and disgust. That's almost inevitable. And then I want to show you a picture of the actual Elephant Man, Joseph Merrick. Here he is. You see that the makeup artists in the movie decided to go a somewhat different route, partly for practical purposes, but you can see the enormous size of his head, the growths of the bone, skin, and connective tissue, and also the fact that his right arm and hand are overgrown and malformed, as were one of his legs principally. His other hand, his left arm and hand are normal. We did not know, or they did not know at the time of his life, he was born in 1862 and died in 1890, what he had. But we now know he suffers from something called Proteus syndrome, which is named after the Greek god Proteus, who was a shape changer. And Proteus syndrome is caused by a mutation in a gene that codes for a specific protein, a macromolecule, an enzyme. And when this mutation happens, it only occurs in certain cells. It's what's known as a somatic cell mutation, and it causes a phenomenon called somatic mosaicism. Somatic mosaicism. So not every cell in his body has the mutant gene. And as a consequence, parts of his body are perfectly normal. But the cells that have the mutant gene fail to control cell growth, cell proliferation. And so he has lots and lots of cells growing where they shouldn't, and you end up with these malformations. It's quite rare. There have been only about 200 to 400 cases reported in the medical literature ever. It's progressive. You're born looking normal, but it starts with birth and progresses through life. And it's of course enormously disfiguring. It does not affect cognition. And as you know from the movie and from the real life of Joseph Merrick, he, thanks to Treves and the way they treated him in the hospital, rescuing him from being a sideshow freak, which is what he had been before, he lived a cultured and sophisticated life in Victorian England and was otherwise normal, but had this incredible disfiguration. So finally, another clip, another still from the movie. The fear we see on seeing someone deformed like Joseph Merrick has an underlying biological basis. That's what we're talking about tonight. And you now understand it. And I hope I've conveyed to you the complexity of that and of the biological systems that underlie fear and other emotions. And you should now be well positioned to understand what causes fear, what creates fear, the physiology, and so on. But these physiological processes also allow us to extinguish fear. So arguably, this ability to feel fear and to learn about it and feel less fear is an example of what we call a homeostatic regulatory mechanism. It's an example of what physiologists call homeostasis, the body's ability to maintain and regulate the physiological, biochemical, and even emotional states. And that's what we do. And all of us have the ability to do this. But we also know that fear can be uncontrolled. And when it is not, when we are not capable of maintaining our fear of the other, of the alien, of the frightening, it can morph into anger. And when it does, we all know from our knowledge of history, of the present, of the 20th century, and so on, we know that the historical consequences of the failure of this kind of homeostatic control can be catastrophic and truly terrible. I want to thank you for your attention. I know I had some technical stuff in there, but I was asked to do the science of Monster, and I decided science. Science is sometimes scary. I hope you weren't scared, but if you were, you now understand what's going on a whole lot better than you did 40 minutes ago. Thanks again. I would be happy to try to answer questions. I'll stop the screen share. I think, Brennan, you're going to maybe help me moderate any questions that come along.
Sure. If you want to type questions in the chat, that's great. If you'd like to use a microphone, I think that's also doable.
That's also great. With this number of people, we should be able to do that. Okay, there's some in the chat.
Yes. Seth is asking what makes scary music scary. So not the visual stimulation.
Yeah, that's a really good question. And I don't really have a good answer, but there clearly are tones and modalities that have been relied on for a long, long time in horror movies and scary movies. And they seem to be often minor modalities. And we also know from some physiology of music studies that minor modalities typically induce different emotions in people than major modalities. And those emotions are usually darker. Those are usually emotions of depression, perhaps of anxiety. And all of those kinds of emotions are tightly linked to one another. Fear, depression, anxiety, which is why we have pathologies that involve those conditions. Let me just give you an example, not from horror, but from musical literature that I'm familiar with. I studied cantorial singing for many years and sang for synagogues, and many songs in the cantorial liturgy and in Jewish folk music even are in the minor modality. And this is because they are sad songs and have a depressing component to them. So I think there's a lot of that going on. The other thing is that certain frequencies are perceived a little bit differently. The way the ear does that is that there's a region of the inner ear which is sensitive to specific frequencies. You have what are called hair cells, which vibrate at specific frequencies and send that signal to the acoustic part of the cortex for interpretation. And some of those, I think, are, when they are triggered, they also connect to and evoke other emotions. That's a bit of a wobbly answer, but that's the best I can do. But it's definitely true. There is scary music and there is not scary music.
Peter is asking what happens in PTSD to make fear response overreactive.
Yeah, there have been studies of that. I did not include them in the presentation. But there is, of course, a lot of interest in that because we have a lot of people, particularly military veterans, suffering from PTSD. There seems to be, there isn't physical damage to their brains as a rule. There can be, but there doesn't have to be. But there seems to be problems with that regulation of excitatory and inhibitory control in the amygdala. There are definitely amygdalar components to that. But PTSD is very, very complicated, and the fear and anger in those people is being routed, I think, principally through the high road. There's cortical involvement, so it's quite complex. But it does involve a dissociation in the parts of the brain responsible for, let's call it fear extinction or fear control, and a decrement in that function. But it's being heavily studied right now, and that's a good thing.
Kirina is asking you to clarify the roles that the temporal lobe and the amygdala play when it comes to fear.
Okay, the temporal lobe, one of the lobes of the cerebral cortex, subsumes certain important functions. Some of the speech and speech understanding components of the cortex are located there. And so when fear is being processed, again, through the high road and cortically, then you're going to see signals coming from the central nucleus of the amygdala out to the temporal lobe of the cortex, where there are lots of centers, lots of important groups of neurons that are responsible for both sensation and also activity.
And Katharina is asking if the editing that we see in horror movies are scientifically informed, or what do they base their choices on?
So if I understand the question right, so are you asking the things chosen in horror movies to scare us, are they chosen with a scientific understanding of the fear response? Is that about right?
Yeah, I was basically just wondering, because I'm in an editing class now, and we were editing horror a few weeks ago, and it seems to me that I'm seeing a lot of connections between the sort of rules that we were taught, but now I'm like understanding the actual science behind them. So I'm just wondering if you think people are actually aware of the science behind them, or they're more
Wow.
so going off of the results.
Great
That they're-
question. And great question. And I'm deeply gratified that worked, because I don't actually know those rules. I'm not a film student, and I enjoy some horror, but like I said, I'm a bit of a wuss. I'm guessing the vast majority of horror film directors are not that clued in to these details, but they know from their own experiences though what makes them afraid. And we have fear memory. So we know certain sounds, we know certain timing of events, we know jump scares versus anticipatory fear. I think the good ones, the Wes Cravens, maybe maybe even some of the B movie ones that worked well, they understood this intrinsically, or they had an intuitive understanding that this is what makes it scarier. One of the things that you see in some horror movies is the juxtaposition of a normally non-scary element, and it becomes Chucky the doll. Although I think he's scary, anyway. But the clown Pennywise and it. Clowns are not scary. Clowns are happy and fun. They stimulate different emotions. But when you are the clown in it, you are incredibly dangerous and scary. And that amplifies that fear. So I think good horror directors, the ones who are successful, they may know some of this science. I don't expect them to know a lot of it, but they understand from their own experience with fear and their intuitive grasp of I think, visual and acoustic art, what they ought to be doing. But I'm deeply pleased that this made sense in the context of the rules you were learning in your editing class.
So
Good.
maybe we can take one more question. Marina is asking, what are the physiological reasons people are attracted to scary things like horror movies?
Yeah, that's a really, really great question. I didn't go into that, but I'm happy to answer the question. The connectivity I talked about in the brain that involves these different neurons that release different chemicals. I talked about GABA, I talked about glutamate. I didn't talk about dopamine. But dopamine is another one of these neurotransmitters. Many of you know it because it's what's lacking in patients with Parkinson's disease. The region of the brain called the substantia nigra is damaged. They can't produce dopamine in that region, and that region is involved with the ability to control their motor activities, so they have tremor. Dopamine is also the neurochemical involved in the pleasure response, one of them, and also with what the kind of pleasure response or satisfaction one sees with certain kinds of drug taking, actually. And in the fear response, dopaminergic regions of the brain can be activated. And when they are, we feel pleasure. So this is the funky paradox of horror and horror movies, is that for many of us, not me so much, but for many of us, it is deeply pleasurable, even though there is simultaneously fear. So it's not having one emotion, it's having multiple emotions, because other regions of the brain will start to get signaling that's coming through the amygdala, coming through the thalamus, and triggering the release of dopamine in those regions, and you'll feel pleasure. A quick personal example: lots of people enjoy roller coasters, even though they're scary. That's also a fear pleasure environment. And I haven't been on a roller coaster since I was 16 because my friends took me to Playland at Rye outside of New York City. And we got on the roller coaster and they're having the greatest time of their lives. And they're pounding on my back. And I was rigid with fear. My eyes were wide, my mouth was open. I could not scream. I was gripping the bars and with basically a spasmodic grip that was not controllable. I lost control of my somatic nervous system to some extent. And when I got off the ride, my friends were laughing and having fun. And I said, I'm never going on a roller coaster again. And that was 55 years ago. So they got a dopamine fix from that ride. I did not.
Jeremy, maybe to wrap up, people loved your presentation, so thank you so much for that. And Veronica is asking if she could have access to the presentation. If someone emails you, could you share?
Yeah.
My understanding is yes, it's very doable. My understanding is this has been recorded.
Yes. Can I just say, something very, very quickly? I am actually sharing with all of you in the chat the link, and the recording will be available probably, I think, next week. So you're welcome to go there and actually see the recording and use it to at your own leisure. And yeah, we're very happy that you are interested in the presentation.
Great.
One last thing I just want to mention, Theresia the Broom, who is somewhere here, is not only ACTI that is sponsoring these events, but also the Marymount Institute. I'm sorry, Theresia, that I didn't that I forgot and spaced out and didn't mention the Marymount Institute. And this is a co-sponsoring of ACTI and the Marymount Institute.
Thank you, everyone.
Yeah, and Theresia's talk is actually tomorrow. So as I said in the beginning, if you're I was going to say glutton for punishment, but if you're a connoisseur of these kinds of things, you can tune in tomorrow and then again on Wednesday and Thursday, and for the other presentations. And they will be very different, but they will be really, really interesting. And I urge you to do that. So thank you again for your attention, and have a great Halloween.